Good morning, good afternoon, or good evening, whatever you are. My name is Gustavo Escola. I am the Research and Funding Specialist at the Canadian Stem Cell Network. While we meet today on a virtual platform, I would like to begin by acknowledging the Indigenous peoples of all the lands that we're on today. The work of Canada's Stem Cell Network occurs from coast to coast and occurs on the traditional, ancestral, and unceded territory of many Indigenous peoples and nations, including the Anishinaabe Algonquin Nation, where SCN is headquartered. SCN owners recognizes and respect these nations as the traditional stewards of the lands and waters where we work and meet. I am pleased to welcome everyone to our third session of the Global Regenerative Medicine Trainee Speaker Series, hosted in partnership with the Australasian Society for Stem Cell Research, the German Stem Cell Network, the Japanese Society for Regenerative Medicine, the Belgian Society for Stem Cell Research, and the Korean Society for Stem Cell Research. We would like to thank a dedicated committee of trainees drawn from each country for helping to organize the sessions and select speakers. The next session of this series is currently scheduled for January. So if you are interested in attending future sessions, please check out the Canadian Stem Cell Network website to see more details and register. We will drop the link in the chat soon. I also encourage everyone to check out the training section of SCN's website and also your home country's network or society webpage to learn more about other training opportunities. I just have a few housekeeping notes before I pass it over to our trainee committee members to facilitate the session. The session will be recorded and posted publicly on SCN YouTube channel. Each speaker will have up to 15 minutes to present and then a few minutes for questions. We encourage you to put your questions in the QA chat box located at the bottom of your screen. Your participation in the discussion is highly valued part of the presentation experience for trainees, so please feel free to engage. Closed captioning is enabled for session. You can choose to show or hide the automated closed captioning. With that, I'll hand over to our first trainee committee representative, who is from the Japanese Society for Regenerative Medicine, to introduce our first speaker. Nahirito Nagoshi, over to you.
I'm very happy to be here with you guys. So where are you joining us today? So I'm delighted to introduce the first speaker. The first speaker for this session is Dr. Shogo Hashimoto. Shogo Hashimoto is a currently assistant professor at Keio University School of Medicine, Department of Orthopaedic Surgery in Japan. And he will be speaking about their work on regenerative medicine strategy for chronic spinal cord injury. So Dr. Hashimoto, please go ahead and begin your slides.
Thank you very much for introducing me, Dr. Nagoshi. I'm Shogo Hashimoto, Keio University in Japan. I'd like to talk about the regenerative medicine strategy for chronic complete spinal cord injury. This is the COI disclosure information of this study. Several therapeutic interventions have reached the clinical stage with the development of basic research on spinal cord injury. For example, we recently performed a first-in-human clinical trial on human iPS cell-derived neural cell progenitor cell transplantation. However, most of this research have involved intervention in the early phase of the injury. Most SCI patients experience chronic severe injury. Thus, treatment for the chronic stages of SCI are fundamentally important. From previous chronic phase of SCI study, cell transplantation single therapy is unlikely to be effective in restoring function because of various inherited factors in decreasing in the survival rate of transplanted cell. Therefore, it is important how to improve spinal cord microenvironment before cell transplantation by combining various treatments, such as a growth factor, rehabilitation, scaffold. There have been no papers reporting recovery of motor function in chronic phase complete injury because of a number of factors that contribute to poor neural regeneration. The difference with incomplete and acute injury is that the residual neural cells and axons are not present along the lesion area, and there is a large cavity with vascularization. On the cavity, there are scar formation consists of fibrous scar tissue and astrocyte, which physically inhibit axonal extension. In addition, the scar tissue secrete a large number of axonal growth inhibitory factors, and chemical factors also inhibit neural regeneration. In terms of inflammatory response, inflammation after injury also persists in the chronic phase of which decreases the effect of neural regeneration and cell transplantation. So to overcome these severe multiple conditions, multifaceted treatment that address each inhibitory factor is needed. We consider the strategy for each inhibitory factor of neural regeneration. First, cell displacement therapy, such as a neural cell transplantation, is a good way for defect residual neural cells and axon along the lesion. In addition to cell displacement therapy, we suggest that improvement of microenvironment at the lesion is essential before such a therapy. As a specific method of microenvironment improvement, scaffold can be formed cavity in a vascular area. Fibrous and glial scar can be treated with scar reduction factor, such as a GHBC or surgical excision. And axon regrowth and neurotrophic factor can regenerate the injured axons. And anti-inflammatory factor can reduce some inflammatory response to improve toward neural regeneration. We consider that it is very important to modify these conditions before cell transplantation. To overcome this severe chronic situation, we have developed the surgical two-stage combination therapy. Here we focus on collagen scaffold, which is used in the clinical practice for artificial damage, which has the ability of enhancing cell viability, promoting vascularization, anti-inflammation, and can prolong the effect drug release. Additionally, we focus on hepatocyte growth factor, HGF, which is a growth factor being applied for clinical trial of acute SCI. HGF has the effect of promoting vascularization, releasing and inhibiting scar formation, and anti-inflammation. We hypothesize that the synergistic effect of combining these materials modifies the chronic microenvironment. After that, human iPS cell-derived NSPCS transplantation enhance functional recovery by formation of new neural circuit. And this is the purpose of study. Can this combination therapy enhance functional recovery in the chronic complete SCI? First, we confirmed with the HGF releasing scaffold modulate pathological change in the chronic microenvironment. Scaffold implantation was performed at 42 days after complete SCI in the rat model, and the analysis was performed at 7 days after scaffold implantation in the 3 groups. No treatment control groups, only scaffold implantation groups, and HGF scaffold groups. Here is the result. Immunostaining for the vascular marker, LECA1, shows that this positive area around the lesion was significantly larger in the HF scaffold groups. It suggests HF releasing scaffold promoted vascularization. Immunostaining for the regenerative axon marker, phosphorylated GAP43, shows that this positive area around the lesion was significantly larger in the HF scaffold groups. It suggested that HF releasing scaffold promoted axonal regrowth. Next, to gain more clear insight into the ability of HF releasing scaffold to improve the microenvironmental conditions, we performed a single-cell RNA-seq analysis by using the lesion tissue 7 days after scaffold implantation. First, 9 cell types were identified by clustering and annotation with UMAP. Comparing the percentage of cell types between 3 groups, we found that microglia and macrophage increase in the HF scaffold groups compared with the other 2 groups. To evaluate which cells are affected by HF releasing scaffold, we analyze the expression of HF gene and its receptor, MET receptor, in each cell. Within microglia and macrophage, we observed a most increase in its expression. In addition, in microglia and macrophage in the HF scaffold groups, we found an increase in HF genes and AR genes, which indicate neuroprotection, and a decrease in the TGF-beta gene, which is scar induction factor. However, BBB score, which is an evaluation of rat locomotor function, showed no improvement in the motor function by HF releasing scaffold therapy. To summarize the above result, the HF releasing scaffold successfully modifies the spinal cord microenvironment after chronic transaction SGI. However, this intervention alone did not improve locomotor function in the SC rat model. We consider that the reason for this was absolute lack of neural cells and axon in the lesion area. Therefore, to compensate for neural cell in the regenerative lesion area, we perform cell transplantation therapy by using human iPS cell-derived neural cell and progenitor cells, which is established in the treatment of SGI, combining with HF releasing scaffold. And this is a method of cell transplantation therapy combined with HF releasing scaffold. iPS cell-derived NSPCS was transplanted at 7 days after scaffold implantation. We transplanted a total 1 million NSPCS at 3 points around the lesion. The analysis was performed at 42 days after transplantation in the 3 groups: only HF releasing scaffold groups, only transplantation groups, and combination groups. Here is the result. First, nearly 70% of grafted cells were differentiated into neurons, and there was no sign of tumorigenesis. Furthermore, the result of measuring human cell volume shows that the combination therapy enhanced the survival rate of transplanted cells. Regeneration of axonal fiber in the lesion area was assessed by immunostaining. Quantification of neurofilament H-positive area, which indicate axon around the lesion, showed a predominant increase in the combination groups. And this image around the region in combination group shows that axonal fiber observed extending beyond the damaged area. In the evaluation of transplanted neuron, neural fiber-derived transplanted cells were observed at extended rostral and caudal transplanted site in the combination groups. To qualify the phenotype of regenerative neural tract, we performed retrograde AAV tracing, which detected only neuron extending beyond the region from rostral site in the combination group. We injected AAV at lumbar enlargement at 42 days after transplantation, and then we analyzed immunostaining for EGFP with rat and a human neural marker at 28 days after AAV injection. The results show that we could observe the regenerative axon-derived host and transplanted neuron extending beyond the region. And many of these regenerative host neurons were found to be propiospinal neurons. To confirm the synaptic formation between host and transplanted neuron in the combination groups, we performed immunoelectron microscopic analysis. The results show that synaptic formation from gold-labeled transplanted neuron to host neuron, and also host neuron to transplanted neuron, it suggested the formation of a new neuronal circuit. As a result, the BBB score, which indicates locomotor function, was significantly improved in the combination groups. We also assessed whether the combination group improved urinary function at 42 days after transplantation. The results show that the maximum bladder wall thickness was significantly decreased in the combination groups, it suggesting the recovery of urinary function. Here we demonstrated that this combination therapy restored locomotor and urinary function even in the chronic complete SCI. And we also proved their mechanism by using several analyses. Environmental modulation before cell transplantation by using HF-redefined scaffold played crucial role in the beneficial effect of cell transplantation therapy. These modifications increased the viability of transplanted cell, which played role of cell scaffold. Furthermore, these cell scaffolds promoted the regenerative of host neuron and extension of transplanted neurons. And these regenerative neurons enhance synaptic formation, it resulting in the functional recovery. Our study also provides evidence for possibility of regenerative medicine strategy by using all clinical relevant materials. We would like to further develop this study for clinical application. We appreciate for all those involved in the study. Thank you very much for your listening.
Thank you, Dr. Hashimoto, for your excellent work and congratulations for your good achievement. This was a nice overview of your work, and I'm very excited to see what comes next. So now we move forward to the question time. So please put your question on the chat, and I will read it to the speaker. So thank you, Cameron. The first question is, are the protocols used here using GMP qualified media and reagent? If not, do you think these protocols can be efficiently adapted to xeno-free growth conditions for clinical applications? Dr. Hashimoto, do you have any answer for that?
Yeah, it's very difficult question. I think this clinical application is a very difficult to make. So There are many steps that we should confirm for realizing clinical application. First, this study was used a rodent model, so that we needed to investigate the effect of the combination therapy for primates, such as common marmoset. And second, we also should enhance more effect of functional recovery because a rat in the combination group in this study didn't work with a rod. And in the future, to enhance the effect of restoring function, we will develop the NSPC transplantation, which is more effective or combined other therapies such as rehabilitation or GHBC, such as, so on. Thank you.
Yeah, because I'm his mentor, so I'll add some comments. So he used three materials, trophic factor HGF and human-derived iPS cells, all of which were already adopted in human clinical trials. In addition, the scaffold, which was commercial available, and it is also used in the clinical setting. So I think three materials can be applied for the clinical application in the future. Thank you, Cameron. So do you have any other questions through chat? Okay. So thank you very much, Dr. Hashimoto, for your excellent work. So now I will turn my role to Cameron to introduce the second speaker. Thank you.
Amazing. Thank you so much, Narihoto. My name is Cameron McKnight, and I'm happy to welcome the next speaker on behalf of the Australasian Society for Stem Cell Research. Our second speaker for this session is Dr. Grace Lidgerwood. Before I introduce Grace, we would like to also acknowledge the traditional custodians of the land we greet you from today, the Wurundjeri people of the Kulin Nation, and pay our respects to any Aboriginal and Torres Strait Islander peoples here online with us today. Dr. Lidgerwood is a mom greeting us today while still on maternity leave, and an early career research fellow in the Department of Anatomy and Physiology at the University of Melbourne. She has an interest in studying the molecular events that lead to retinal degeneration, including macular degeneration using human-induced pluripotent stem cells. Today, she'll be speaking about her work on translating patient stem cells into personalized screens for age-related macular degeneration. Grace, please go ahead and begin your presentation.
Thank you. I think I might need Shogo to stop sharing screen, or can I override that?
I think you can, yeah, there you go.
All right, is that looking okay?
Yes, great. Take it away.
Okay. So thank you very much for the invitation and for the organisers for the opportunity to present here today. So I'll be talking to you about our work on macular degeneration using a large-scale stem cell approach to disease modelling. So age-related macular degeneration, or AMD, is the leading cause of blindness in the developed world. And this number is predicted to grow as our population ages. It's a disease that's characterised by death of the retinal pigment epithelium, or the RPE cells, and this leads to the death of the overlying photoreceptors in a region of the retina known as the macula. And the macula is responsible for central vision. So if you obviously have loss of cells in this region of the retina, you will lose your central vision, and this obviously has an enormous morbidity for patients. So AMD typically progresses slowly over many years, and it can progress to one of two forms of the late stage of disease. The first being geographic atrophy and the second neovascular AMD. So we know that anti-VEGF injections are a useful strategy for dealing with the latter form of the disease, but most patients actually go on to develop the geographic atrophy form. And to date, there's still really no effective therapy despite a lot of international focus and research on this disease. So we know that there are lifestyle and risk factors that increase the likelihood of developing the disease, but there's no one definitive cause. And this really highlights the need to us for greater focus on developing reliable models of the disease. So human pluripotent stem cells have obviously been used really successfully to study monogenic diseases, but they can also be used on a much larger scale for complex heterogeneous diseases like AMD, which are more challenging to model because there is no one specific underlying cause. So for disease modelling of this nature, we require really large sample sizes to be confident that we're accurately modelling and representing the disease, and also giving greater power to the study and the analysis, the subsequent analysis. So we took this approach and we established a large-scale model of geographic atrophy using induced pluripotent stem cells. And so in the end, we had 43 GA lines and 36 controls that were differentiated to RPE using our established protocols. This process takes around 90 days for the cells to reach full maturity, and they're distinguishable really easily because they actually turn pigmented, so they turn black in the dish. But we also obviously performed rigorous quality control to ensure that the differentiation was successful. So we're just demonstrating some canonical RPE markers here, and we did a number of other quality control measures to be confident that we had RPE cells. And at this point, we flew the cells up to Sydney with our collaborators at the Garvan Institute, who took over the single cell sequencing and EQTL analysis. And also simultaneously, we took some protein samples to our collaborators at Macquarie University, also up in New South Wales, for the large-scale proteomic study. And so taking this multi-omics approach provides a really rich data set that we hoped would provide in-depth information about some of the genetic and molecular aspects of the disease. And so what we found was that there were several subpopulations of RPE in our disease model. The progenitor population was very, very small, which we expected. It accounted for less than 6% of all the sampled cells. And these are not yet RPE, but they have progenitor RPE markers, which commits them to becoming RPE cells. But by RPE population 1 and 2, they had reached a mature RPE phenotype, which we're just showing here, demonstrating here with some of the canonical RPE markers. And then they continue to mature as they move through populations RPE 3 to 6. And this really essentially confirmed kind of what we already knew, was that the samples that we sent off were indeed RPE cells, so a tick there. And that's important for a number of reasons. Because the RPE cell is the cell type that's affected in disease, obviously we want to be sure that that's the cell that we're actually sampling in our disease model. And from the sequencing data, we wanted to answer two main questions. So what diseases are, what genes are differentially expressed? So is there an association between genes being more highly upregulated or downregulated in disease? And secondly, how is gene expression regulated by genetic variants? And the way we can answer this is by examining EQTLs, which are part of the DNA, or usually SNPs, that regulate the expression of other genes. And they can be measured quantitatively using computational biology approaches. And importantly, EQTLs are cell type specific. So I guess that really drills in the importance of confirming that we were sampling RPE cells for this disease model. So focusing on the differential expression analysis, we found that there were more than 3,000 genes that were differentially expressed between GA and control samples. And 27 of these had known and published associations with GA. And then they're shown, the significantly differentially expressed are shown in the red boxes on this dot plot. And this is really exciting because essentially it showed that our model was sensitive and accurate enough in picking up genes that have already been identified in previous studies, but also importantly, it identified new hits that haven't previously been associated with the disease. So when we analysed the biological pathways that these differentially expressed genes fell into, we found that they were significantly overrepresented in processes involved in mitochondrial pathways. So ATP metabolic processes, the assembly of the respiratory chain complexes, oxidative stress, and so forth. And we also found that regulation of extracellular matrix protein, extracellular matrix genes was altered, which we know is a key feature of the disease. So in almost all cases, the genes were upregulated in the GA cohort, suggesting that there could be a key difference in the energy demands of the cells in disease and also in the remodeling of the extracellular matrix. And the results of the proteomic screen also pointed to major changes in mitochondrial pathways. Again, these were mostly upregulated in the GA samples, and they were most often associated with the assembly of complex 1 and ATP synthase, but also a number of other different, I guess, ontologies that these fell into, which are shown here. So oxidative stress pathways, mitochondrial functions, ECM regulation as well. So we found that the single cell sequencing and the proteomics data really complemented each other, which was really exciting because we know this, not always the case. But we wanted to take it a step further by looking at EQTL and PQTL analysis. So when we looked at the EQTLs, we found that there were 445 unique EQTLs associated with the disease, and many of these have previously been identified. So an example of this is PIL-RB, which is an example of an EQTL, a gene that's regulated by an EQTL across a number of populations in our analysis. Oops. But we also found new EQTLs that haven't yet been linked to the disease, and many of these were also involved in mitochondrial processes. So I've outlined these in some of the red boxes here. And so again, this points to some really interesting differences in mitochondrial profiles in EQTL regulation of gene expression between disease and healthy patients. So as protein expression doesn't necessarily always correlate with mRNA expression levels, we performed a cis protein quantitative trait loci or cis PQTL analysis to identify genetic variants that regulate protein expression in the RPE in the context of disease. And we identified 5 proteins that share a lead SNP with RPE subpopulation level EQTLs. And 3 of these, which is PyroxD2, SPATA20, and crystallin, was significant. And 2 of these are known to regulate the expression of proteins involved in mitochondrial function. So again, this really validates or strengthens this theory that mitochondrial pathways are altered in disease. And to the best of our knowledge, these were the first RPE-specific PQTLs to be associated or identified with AMD. And what we wanted to finally do was validate a lot of this functionally by demonstrating mitochondrial changes in a semi-functional assay. And so we analysed a range of parameters in oxygen consumption rates between GA and control RPE using a seahorse analyzer. And what we showed was that maximal respiration and spare respiratory capacity were significantly reduced in the diseased samples. And this data essentially, I guess, crudely validates many of the significant findings from the omics approaches, although we understand we do still need to do a lot more in-depth analysis to understand the deeper functional components of the disease. But it seems to really all complement each other, which was really exciting. So I guess just to conclude, our study was able to successfully model end-stage AMD, so geographic atrophy. It was able to predict and detect previously identified hits from other studies, but also importantly, it yielded new results that haven't yet been described in the disease. And we have strong evidence for major differences in mitochondrial pathways in the disease. And we're now hoping to screen our GA lines with large mitochondrial compound libraries to hopefully identify therapeutics that might restore mitochondrial function and rescue the phenotype associated with that. We're actually currently undertaking a large-scale study of this type, just not on GA, on a different macular disease, and we've found some really exciting results in that space. So I guess that's just to sort of roughly sort of summarize where we'd like to go from here. So we'd like to understand in more detail. So looking at other functional assays, potentially looking at knockouts, knock-ins, going into more detail. So looking at ATP synthesis assays, ROS assays, and really starting to understand this in much more detail. So the work was an enormous effort by a large number of people across Australia and more broadly. These are just some of the names of the people involved in the study who were fundamental to the work. So thank you to that, and thank you to all the funding sources. And yeah, thank you again for the opportunity to present here today.
Amazing. Thank you, Grace, for a great overview of your work. We really look forward to seeing this progress further. So we have time for a few questions from the audience. Please put your questions in the Q&A box if you have any. In the meantime, I might start us off. I know you have a lot of follow-up to do for the mitochondrial work, and I'm excited for you to get back to that. But I was curious, in terms of drugs that you think might be promising, are you going to be looking at a targeted set of mitochondrial specific drugs, or are you going to go with a compound library?
So ultimately, we'd love to go with a compound library. We had to start, I guess, small first to then to make sure that our model, our approach is feasible, I guess, on a large scale. I think our library that we wanted to look at was about 200 mitochondrial targeted compounds. I think before leave, I narrowed that down to maybe 10 or 20, which we've just screened on a different study. But obviously the volume of plates that you need to do a compound screen on a large scale disease model is enormous. So for that particular study, we obviously started small, optimized all the parameters, optimized our readouts of whether the drugs were actually having an effect on the disease phenotype that we were able to measure. So I purposefully sort of went through the literature and really selected what I thought would be appropriate candidates for that compound screen. A number of them were antioxidants, which I think we've actually had some really interesting results with that so far. I won't talk too much about that, but yeah, so we had to start small first. But the aim is, once we've got this platform and this pipeline really, completely optimized, the aim would be to then extend out and screen for more compounds.
Amazing. I think that's a great approach. I mean, as a mitochondrial person myself and doing large scale screens myself, I know that generating these cells from IPS is a huge demand, especially particularly laborious. Do you guys have a scalable protocol, or is that something that needs to be built up in order to do larger scale things?
No, the protocol is pretty it's pretty reproducible. I mean, it can be a little bit fiddly in terms of line-to-line variability for optimal density of seeding initially, but once you've really optimized that step, the first 7 days of the protocol, it really generally does work quite well. It doesn't involve cutting, it doesn't involve 3D formation of any cells. It's just a monolayer, and if it's done properly in the first 7 days, it yields really beautiful monolayers of RPE cells. It is scalable at the moment. We are doing it manually just because we can, but it definitely is scalable, and we've used robotic platforms in the past to sort of create an automated approach to disease modelling. We just haven't done it for the RPE projects, but yeah, the protocol lends itself really nicely to automation and robotics, I guess. Yeah.
Amazing. Well, it's great to make sure that you think all the way to the end of these projects. So it's great to see that you've done that. Yeah, well, I'm going to stop there, and thank you, Grace, for your presentation. We encourage any interested audience members to contact Dr. Lidgerwood and keep the conversation going. So I'll now hand it over to Sang-Hoon Park to introduce our next speaker.
Yeah, thank you, Cameron. My name is Sang-Hyun Park, and I'm based in Korea. I'm excited to welcome the third and last speaker of this session on behalf of Korean Society for Stem Cell Research. Our final speaker, Ju-Hyun Lee, is currently a a senior researcher at Korean Institute of Science and Technology, and will be speaking about their work on production of human spinal cord organoid, recapitulating neural tube morphogenesis. Ju-Hyun, please go ahead and begin your presentation.
Yes, can you hear me now?
Yes.
Yes. Thank you for kind introduction, Dr. Park. Good morning or good afternoon, everyone. I'm Ju-Hyun Lee from the KIST Brain Science Institute. Before I begin, I would like to thank the coming member of this seminar. It is my great honor to be here and to have the opportunity to present my research. Today, I will talk about my research on human spinal cord organoid and their role in modeling human neurodevelopmental disorder. Yes, there is no doubt that brain organoids are a valuable model in neuroscience. On this slide, you can see an overview of key application and limitation of the current organoid technology. First, the green circle on the left represents the key application, including human developmental biology or human disease modeling. First, in particular today, I want to emphasize the drug test field. The organoid-based drug screening has become increasingly significant because of the recent global policy of banning animal experiments. However, before we can start using organoid-based drug screening, we need to address some weak points. One of them is the absence of the robust quantitative analysis system, especially in case of brain organoid. It is more difficult to overcome this issue because of their complex structure and high heterogeneity. So my project goal was to make spinal cord organoid with a focus on this issue. Yes. To achieve this project goal, I have several key strategies. The first strategy involves mimicking the process of spinal cord organoid, a spinal cord development in vivo system, as other organoid researchers have done. For that, we adopted a novel theory proposed a few years ago. Before that, we have believed that the entire central nervous system originated from the anterior neural plate. But however, the new concept suggested the posterior part of the central nervous system, including spinal cord, had a different origin called neural mesoderm progenitor cell. With this novel theory, we have tried to make the spinal cord organoid derived from neural mesoderm progenitor cell, not anterior neural plate. And second strategy was to use 2D early induction method instead of 3D. As I mentioned before, brain organoids have high heterogeneity, causing little progress in quantification assay. To address this issue, we decided to optimize from the initial stage of the procedure. In order to minimize cell heterogeneity from the earliest stage, we adopted a 2D culture method to induce neural mesoderm progenitor cell. This approach results in more unidirectional cell differentiation expressing SOC2 and brachial teeth compared to the 3D differentiation method using embryonic body. Furthermore, during the 3D conversion process, we used the reaggregation method following cell dissociation to enhance reproducibility. In summary, by fine-tuning the initial step of the protocol, we try to develop organoid model well suited for their application. So with these two strategies, we started to generate this spinal cord organoid model. Interestingly, During the 3D cell culture system after 2D induction, these organoids show specific morphological change under the bright field microscope. So we attempted to observe each stage in detail using 3D format staining. Please refer to this video below. It was clear that they were undergoing the morphological alteration. In addition, it looked like some in vivo neurulation process. Yes, so to examine whether these organoids recapitulate the neurulation process, we performed the immunohistological study. Compared to in vivo neurulation, they show quite similar feature consisting neural plate and neural folding and neural tube closure. Additionally, when we zoom in folding region, like that, in this region, we could observe ventral hinge point cell formation and planar cell polarity arrangement having medial lateral arrangement and zipper-like closure of neural tube. All of these aspects show similarity to neural folding process in vivo system. Following the termination of morphogenesis, the organoid returned to round shape, and the elongated neural tube was located in the organoid. Furthermore, they exhibited typical in vivo neural tube feature, including intercalating nuclear migration and presence of mitotic radial glia cell and the primary cilia at apical site. Also, differentiated neurons surrounded the outside of the tube. Based on these compelling results, we concluded that these organoids recapitulate in vivo neurulation process. Next, to confirm the identity of these organoids, we perform microarray analysis using human embryonic spinal cord tissue as a reference. The gene expression profile of long-term cultured organoid has a higher degree of similarity to human embryonic tissue compared to earlier sample. To further address the cell type within organoid, we conducted single-cell RNA sequencing on 1-month organoid sample. These data demonstrate that the spinal cord organoid comprised a diverse cell population, including both mitotic and post-mitotic cell with nodoventral identity. Next, to confirm the maturation of spinal cord organoid, we performed an immunohistological study with several specific markers. As we extended the culture time, the organoid showed a mature astrocyte and myelinating oligodendrocyte. Similar to mouse embryonic spinal cord in panel G and H, carbidin positive interneurons show heterogeneous population, and we observed the evidence of mature neurons showing dendritic pain in panel L and synaptic contact in panel M. And then we performed the microelectrode array experiment to examine the physiological property of organoid. These results show high heterogeneity and synchronized neural activity within our organoid model. Also, we observed the neural firing and bursting rate gradually increase over time, which is a typical feature of the developing spinal cord tissue. So to summarize the results so far, we generated 3D spinal cord organoid model recapitulating many characteristics of the in vivo spinal cord tissue, including neural tube morphogenesis. In the next part, based on this key feature, neural tube morphogenesis, I'm going to talk about their application for biomedical research. This slide highlights the quantifiability and reproducibility of our spinal cord organoid model. To optimize the morphogenesis condition, we adjusted cell number and growth factor treatment duration. These two factors control the organoid size and neuroepithelial thickness in the neuroplate stage. And then we carefully observe and quantify the success rate and speed of their morphogenesis. Most conditions successfully generated neural tube, but some organoids having thick neuroepithelial layer failed to generate the neural tube. It indicates that the neuroepithelial thickness is the primary factor affecting morphogenesis rather than the organoid size. For the application of our organoid, we try to establish a drug screening platform based on deep learning technology. This system included an automated imaging system and AI-based image classification method. Using the acquired bright field images, hundreds of samples could be easily classified into three stages, such as neural plate, neural folding, and neural tube. In short, this system allows the robust quantitative analysis of neuralization process. Next, we examine whether the organoid could be used to model neural tube defect. A neural tube defect is one of major congenital malformation caused by abnormal neuralization process. Clinical studies have consistently shown that prenatal exposure to anti-epileptic drug is associated with neural tube defect. With this information, we try to study neural tube defect caused by anti-epileptic drug using our organoid model. First of all, we selected 6 types of anti-epileptic drug. These drugs are listed according to order of risk from valproic acid to gabapentin. During the period of neural tube morphogenesis for 8 days, we added these drugs into the culture medium, and we quantified the success rate and time required for morphogenesis. These heat map graphs make this trend easier to recognize. Interestingly, only valproic acid and carbamazepine treated group show significant difference compared to the control. In these two groups, the initiation and termination of morphogenesis were both delayed. And we examined 3D neural tube structure in the drug-treated group, comparing them to the control group. Valproic acid and carbamazepine displayed different shape of tube, which look like opened neural tube. These observations align with the trend in human clinical data. According to these clinical results, only few drugs, including valproic acid and carbamazepine, are associated to neural tube defect. In conclusion, these results demonstrate the potential of our organoid model for drug toxicity test. Because the valproic acid showed the most severe phenotype in the previous assay, so we further investigate the valproic acid induced neural tube defect. These results show that regardless of treatment period, valproic acid exposure could delay the folding morphogenesis. Next, to understand the molecular changes induced by valproic acid exposure, we perform microarray analysis. We found significant change in cell-cell adhesion, especially tight junction protein. To track tight junction expression pattern, we perform immunostaining. Usually, tight junction protein are located on apical side of neural plate or neural tube. However, when we treat the valproic acid in organoid, apical polarity of tight junction was disturbed and show ectopic expression. These results suggested that valproic acid induced neural tube defect is associated with tight junction. In addition, we investigate the mouse model induced neurotube defect by referee acid exposure. Although there are some interspecies differences, we observe the abnormal expression of tight junction protein in animal model. So, summary, we generated the human spinal cord organoid model and also established the drug screening platform based on AI technology. And using this system, we studied neurotube defect caused by anti-epileptic drug. This is the last slide of my presentation. All the work was done at my previous laboratory, and thank you for your attention.
Thank you, Ju-Hyun, for the interesting talk, and I can't wait to hear more in the future. We now have time for a few questions from the audience. To ask a question, please put your question in the chat box, and I will read the questions out loud to the speakers. We have a question from Cameron. The question is, incredible work, Ju-Hyun. It is great to see such a robust model of the CNS in vitro. Have you considered running metabolomics and or lipidomics on your organoids to better understand how the maturation is impacting their structure and mitochondrial function? Would you expect the metabolic switch in more mature cell types?
Yes, thank you for your question, Cameron. Actually, we have not experienced about the metabolomic study or lipidomic study, but I'm sure that as a culture time period, maybe metabolism level will be switched because the composing cell type within organoid, the cell type, we already checked the cell type, mainly cell type changes continuously, and their metabolic tone also increase, I think. Yes, I expected the metabolic level change in more mature cell type, including astrocyte or oligodendrocyte.
Okay, thank you for your answering. I'd like to add one more question on my mind. It's really amazing that spinal cord well mimics various stages of neurulation. As you know, during in vivo neurulations, neural crest cells originated when neural tube formation. So I'm wondering if neural crest cells have been observed in your culture system as well.
Thank you for your question, but we already checked the specific cell type, including neural crest cell or mesodermal cell. But unfortunately, in our organoid model, we could not find the neural crest cell. Yes, so actually, I think that is our limitation of our model. The only few type, there are only neural epithelial, including neural epithelial, the neural lineage cell type. We only include the neural specific cell type.
Okay, I understand. And according to transcriptome profile data, it seems that the spinal cord organoids contain cell population with ventral identity, even though your protocol doesn't involve the use of any ventral morphogens like Sonic, but human neuroepithelium adopts those identity by default, as far as I understand. So I'm wondering how they acquire ventral identity.
Yes. Although we did not any sonic or BMP signaling, we can easily find found out the nodalized cell type or ventralized cell type. But it is not too much. But when we treated the sonic hedgehog or BMP, we can induce much better than what we wanted to. So it means we thought in these organoids have potential to induce their potential to induce by signaling factor, but neutral condition in the normal condition we cannot we can find only few type of nodalized or ventralized cell.
Thank you. And I think this will be our last question from Narihito. Do you have an idea to apply your system to regenerative medicine for spinal cord injury? For example, what do you think about transplantation of your organoid tissue?
Yeah, thank you for question. Thank you for a nice comment. But actually now I'm not I don't have any plan to apply the regenerative medicine for spinal cord injury, but because our organoid is too small compared to other organoid research. So for regenerative medicine, maybe the content or quantity of organoid should be increased. So, but as I know, the other researcher already tried to apply the spinal cord injury model using spinal cord organoid model. So I think it can be useful because the spinal cord, yes, and useful because they already published the dramatic result after transplantation. Yes, it will be, yes, I think it will be good.
Okay, thank you. Thank you again, Ju-Hyun, for taking the time out today to share with share with us a little bit about your work. We encourage interested audience members to contact Ju-Hyun to keep the conversation going. That brings us to the end of our 3 talks for this session. I'd once again like to thank our speakers, Shogo Hashimoto, Grace Lidgerwood, and Ju-Hyun Lee for their excellent presentations today. I'd also like to thank everyone in the audience for their attention and all of the networks who are partnering on this speaker series to give a platform to rising star in stem cell and regenerative medicine research. We hope you are feeling inspired because the future is very bright. Please be sure to check out the Stem Cell Network website for upcoming sessions of this series, which will be happening next in January. On behalf of SCN and the partner networks from Canada, Australia, Germany, Japan, Belgium, and Korea, thank you all for attending and wish you well. Thank you.
Thank you all.